Hello, Jane. So we wait for your friend for too long time until you start. Okay, from the last class, we talked about many organelles, right? And we stop at the cell membrane. So at the cell membrane now, I think you know already that it's semi-permeable membrane. Do you know what does it mean, semi-permeable? Exactly, the meaning. That what is the property of the cell membrane. I know you are tired, but just listen. Does it allow any compound to get passed through? All compounds, all kind of compounds? Yeah, which kind of compound that cannot pass through it? Small molecule and non-polar compound can pass or cannot pass? Can pass through. Okay. Right. Yeah, so normally, if you look at the cell membrane itself, it's the biphospholipid, right? Layer. So that means it's two layers of this one is a lipid tail and the head is a phosphate. So normally the compounds can get through this easily, but it has to be small and has to be non-polar as well. Yeah. And if they have the polar, just like ion that dissolve in the water already, they cannot get through it, just like sodium and chloride, like that, because they have the specific channel to get passed through. That's one called carrier protein. That's what your friend said. So the carrier protein can get past all the ion, and what else? Don't talk about gas. It can get through easily. Thinking about bigger protein. Amino is too small, right? Amino acid is the smallest compound in there of the protein. Smallest unit of the protein. So you think about the big protein and what else? Carbohydrate that are very huge. Yeah, so they cannot get through easily. Okay, so this one, that's why it's called semi-permeable membrane, because it can get passed through some of them. Okay, smaller, non-polar, easily, very easily. Why it have to be non-polar? Black. Sorry, I have to say Thai sometime also. You don't mind, right? Just sometime. Okay, for the negative charge at the phospholipid head of that your friend said, where is it? Here. So they can catch some of the compound over there. Of course, but normally the non-polar can get through the lipid here. Just like your skin, look at your skin. Okay, when you put a cream on, they have to be a bit of lipid. So they can absorb easily because it's phospholipid, so some of lipid can pass through easily. But if the compound dissolve in totally in water, you just spray the water on, can the water get through easily? No, right? You have to take some time, yeah, to let it pass through. Just like the water itself, you spray on, right? You just try to put it in, but they cannot get through. So normally the cream, the cosmetics, or the spray, or the lipstick, so they have to be likely to get through our membrane everywhere. And what is the pH our skin? Do you know? Perfect. 5.5 is the acid. Remember, our skin is the acid at 5.5. So if you make the cosmetic that has the same of pH, is the best. So adjust it to be 5.5, but not lower than that because we will irritate to your skin. 7 is fine, isn't it? Because 7 is just the middle of the pH. Yeah, no, nothing can harm you. Just like the water, the pH is 7, so nothing can harm you also. Yeah. And if it's over than 7, just a little bit is fine too. But if it's over than, what's wrong? If you burn your skin as well, just like the acid, the strong acid or strong base can burn your skin. Yeah. How about the washing powder in the toilet? Is it acid or is it base? Base. Yeah. All of them are base, very strong base. That can irritate your eyes a lot, irritate your skin a lot. Yeah. I have my professor that I used to study the PhD before. She is the owner of the, what is that? Do you know Tatayan? Is she too old for you? Yeah, she is the presenter of the skincare. Called what? I forgot the name. I forgot that name. Tatayan that she, yeah. Yeah, so I shouldn't mention it because I recorded interview. Okay, so my professor, she is the owner of that cosmetic factory and that brand. That's very famous in Thailand too. Yeah, it's very huge market. So she is the one who created the negative charge of the cosmetic, just like the first one. She knew that our skin is 5 by 5, so she is the first one in the market who created that. That's why she's popular. All the doctors just came to her and she just produced everything into the nanoparticle to absorb it easily. Yeah. So if you know the trick of your body, the trick of the cell, see, you can make a lot of money from it. So don't say that the basic knowledge is not good. Yeah, if you know all the basic knowledge, you can get wealth from that. See? This one is an example of get well, but we have to come to our class, right? So we have a lot of protein here, and you see that we have a carbohydrate chain that normally attached to our protein too. That is the receptor. They will select what kind of compound that we can get through the cell membrane. So they all are the receptor. So they have the signal on them. So these are very important of the protein, carbohydrate, and lipid on our membrane. Okay. So if you see that the phosphate part is the outside layer, right? So it's like water, of course. So we call it hydrophilic. Philic is love, like, right? And the opposite of philic is phobic or phobia. That means hate, right? So this one, all the lipids are hydrophobic. And inside our cell also, inside our cell are cytosol. And the cytosol mainly is water, isn't it? So they have to be the phosphate. So that's why the phosphate come in here again. Okay. And between the cell, here is between the cell, think about between the cell. This one is the first membrane, and the next one is the second membrane here. Between them are extracellular matrix, or ECM, have a lot of compounds in there. That's very important for our body, just like elastic fiber, collagen, polysaccharide. So they make our skin to be flexible and soft. If you touch our skin, it's not hard, right? It's soft. So because we have the elastic fiber and collagen here. So if you try to feel the collagen, you have to feel at the ECM, not into the skin, okay? Not into the cell, right? And here you can see the receptor protein, and they have the cytoskeleton that attach to protein that try to maintain the shape of the cell. We talked about that yesterday. Okay, you know all the cell membrane. And inside of the cell, we have the main thing that we call nucleus. That's just like our brain. So it thinks and it's trying to control everything inside of the cell, right? To produce all the compounds in there too. And here, the middle of the nucleus, we have the very hard thing. It's not that hard, yeah, but it's kind of solid. We call nucleolus in there. And here, the chromatin is inside the nuclear envelope, isn't it? Here is a nuclear envelope. And nuclear envelope have a lot of pore here. We call nuclear pores. Very little area, yeah. And the chromatin is here. So you cannot see the chromosome until the phase of cell division. So they will contract, they will condense before the cell division only. But normally, they will expand themselves in the chromatin form. And can the chromatin get out, or I should ask, can the DNA get out of the nucleus by itself? No, we have to copy it, right? What we use to copy? Huh? RNA. We copy by RNA. We use mRNA to transcription the DNA into the RNA. And then the RNA will be translated into a protein by tRNA. But the mRNA will copy the DNA inside here. And then they will get through the nuclear pore into the cytoplasm. And at the cytoplasm, the translation will happen. And the protein will be produced there at the cytoplasm, right? You still remember all of this, isn't it? Yes? No? Please. Remember? Okay, what is the full name of mRNA? tRNA? Transcription RNA. And the tRNA always work with ribosome to produce protein. This process we call central dogma. Okay, from the DNA to mRNA and protein, we call central dogma. That means it's a process of DNA to work, just like it's a working of DNA. You cannot just use DNA to function or of your hair color, or not hair color, hair color, or your skin, or your eyes, right? You have to use a protein to express all of that characteristic for you. So you have to translate it into a protein first, then it can work. And here we know that it's transcription and translation process. Okay, it's very easy for that because I just want to remind you, but I want you to see this, notice this. There are others of organelles that attach to nuclear pore. This one, can you see the layer? What is this organelle? Endoplasmic reticulum. Okay, so endoplasmic reticulum, or ER, is a follow-up of nuclear membrane. It's the outer layer of nuclear membrane that fold up together. If it has ribosome to attach, so we will call rough endoplasmic reticulum, or RER. And when it doesn't have, it will call smooth endoplasmic reticulum, or SER. I will come back to ribosome later, okay? Just go to ER first because we talked about that already. So if you look from the electron microscope, you can see here is the membrane of ER. And you can see that a lot of ribosome here that's scattering around. And this ribosome have two units, or we have two units, large unit and small subunit. Okay, that work together. For the RER, what is the function of the ER? Produce protein. Good. Okay. It's good to speak out, if you practice your English also. Don't be shy, just speak it out. Here you will produce the protein, it will produce the protein, but together with the ribosome. Yes. And if so, we call that, it's a lot of them in the organs that have to use a lot of protein, just like liver and pancreas, because they work all the time. Think about that. They work all the time, they will destroy all the time. So they have to rebuild all the new cell all the time too, because they got medicine, right, from our body. So they have to produce energy. That's from which organelle? Mitochondria. Yeah, they have to produce energy for mitochondria. So the liver and pancreas have a lot of mitochondria, and just get along with the RER and ribosome. Yeah, they have a lot too. So we will not emphasize here more, but look at the SER that normally we are not used to them. You know that it's exactly that the rough ER here with the ribosome, but the smooth ER is attached to the rough ER too. So can you imagine that they go along together? Nuclear membrane and then go to RER. At the end of RER is the SER. So what is the function of SER? Produce lipids. Remember, okay. One thing, lipids. And all together with the lipids, it's the same kind, it's a steroid. Yeah, steroid is the lipid structure. So our body have our own steroid. What is the function of steroid? Sex hormone. What else? Meaning? Underneath the kidney? Oh, no. I mean the function of it, the benefit of steroid. Normally, what you use a steroid for? Huh? When you have fever. Yeah, apart from sex hormone. Okay. When you have a fever, you have anti-inflammation. Or when you have inflammation, so you can use the steroid also to stop that inflammation. There are a lot of medicine that are steroid, so it will suppress your immunity, because when you just faced with the stimuli, just like bacteria or virus or any wound, any cut, so you will have inflammation at the spot. Okay, that's inflammation. You have to use the macrophage, right, or the white blood cell to defend them. And if it's overreact, you will get a fever, you get pain and redness after that. That's inflammation symptoms. That's normally. So how can you stop that overreaction? So you can apply the steroid that we use in the old time, that normally use the steroid. But at this time, we try to synthesis another medicine that just look alike steroid. It's called NSAID, non-steroidal, yeah, anti-inflammatory drug. But they have the same anti-inflammation also, but it's not steroid. So the structure are different. We come back to our RER. So inside our body, so we have our innate defensive mechanism that have inside the cell. So we have the steroid from the RER by ourselves. Yeah, to defend that inflammation that happened in our cell. Okay, so this one is the useful of SER, lipid, steroid, and it produces carbohydrates also. But mainly you remember this lipid and steroids normally come from them. And they will detoxify of the drugs and harmful substance for us. And it's used for calcium storage as well for our muscle cell. Okay, we call sarcoplasmic reticulum. This one we will see again in the next 2 chapters. I will teach you about this. Okay, sarcoplasmic reticulum. And also they have another structure that was like, it's a dynamic modified structure, just like in the cytoskeleton. So it will use to just like make our cell more flexible when it's there. You think about the lipid. Lipid is soft and flexible. So when it produces lipid, we make everything soft and flexible too. That's the SER. Yeah. Okay, here, that's how it's produced the protein. Here is the SER envelope, right? And the ribosome will come together with the mRNA and tRNA, and they will capture amino acid and connect each amino acid to be a chain of the protein. And then this protein will fold up together, and then will fold up with the membrane of the ERT. So it will transport to the next organelle as a vesicle of protein. Vesicle means the folding up of the ER into a bag and has the protein inside. Okay, there is a protein vesicle. And it doesn't work only with the protein, see? It will attach all the sugar groups here too. So the protein and carbohydrate always works together. This one they call cisterna. It is the just like the stick, the antenna of the ER, and then they call cisterna, ER cisterna. Sorry. If you attach the sugar at the protein, they will call glycoprotein. Okay, this is the English form. If you just put something at the suffix, that means it's the biggest. Just like glycoprotein means protein is the bigger one, but the glyco is a suffix. It is a prefix, right? It's an adjective of them. So it's just a small amount of glycoprotein, small amount of sugar on the protein. Okay, up until now, do you have any question? In the shape of the ER, it's called like this, right? So each one they call cisterna. Okay, the next of the organelle that work together to produce protein is the Golgi complex, or we can call Golgi body, Golgi apparatus, or whatever. And this is a shipping and receiving center. When they got the protein, they will read and decide where this protein can go or where this protein should go. So when the vesicle get in here, have to be number one. When the vesicle get in here, they will fuse with the Golgi complex first. And then they will attach a lot of things. It's like another protein, another carbohydrate. Yeah, inside here. And they will release with another vesicle from the Golgi complex. And this one will be ready to go to other organelles. It can go to plasma membrane or can go to the other organelles to build up another plasma membrane of other organelles, or can be the food vesicle for other cells. Yeah, or can be export outside of the cell to use in another cell too. So whatever it goes, Golgi complex will decide where it should go. Okay, or if you look at the electron microscope, you can see that here is the Golgi complex apparatus. They will release a lot of protein vesicle here. So it come together, right? Just like the system that we call endomembrane system, that it get from the nuclear envelope, go to the RER, go to the SER, and then produce a protein vesicle, go to Golgi complex, make another protein vesicle, and then go to other organs. Yeah. Okay, come back to the ribosome. Ribosome have two units that I told you that will read all the mRNA together with the tRNA. We cannot see the tRNA here, but imagine that it always come together, okay? So they will read up the protein and then produce here. So this protein will go to nucleus, mitochondria, chloroplast, peroxisome, whatever. This one comes from free ribosome in cytoplasm. Free ribosome means the ribosome that not attached with the ER. Okay, so it's just by itself in the cytoplasm. So they will the protein to these organelles. But the ribosome that attached to the ER, we call membrane-bounded ribosome. They will produce two plasma membrane, secretory vesicle, endosome, and lysosome. Don't have to remember this. Okay, it doesn't matter where it's produced to. Just know that it's produced protein for yourself. Okay. I just told you that there are many kinds of this in our cell, okay, but don't remember anything. Yeah, just in the future, maybe you can pick up this knowledge to use for further study. So we finished about the endomembrane system. Do you have any questions? Cannot follow anything? We almost finished the class. Okay, if you don't have, I will go further. So these chloroplasts that only have that in the plant, there are two kinds of chloroplasts also. The first one that produce pigment, we call grana chloroplast. Okay, grana means they have a granum. It is a granum, right? That is a thylakoid stacked up. So the thylakoid look like a coin, and then you stack up, right? That's one a portion we call grana. Okay, many we call granum. Many of granum we call grana. So this one is a grana chloroplast, produce pigment in there because this thylakoid have a chlorophyll that attach to the membrane of the thylakoid. Still remember that? Don't worry. You will study again in the photosynthesis. Or I will show you later about the picture. Here is a thylakoid, that's what I mean. And here why is green? Because it has a lot of chlorophyll on them, right? A chlorophyll attached on this membrane. Yeah, so it's stacked together to be a granum. And then if they destroy all the granum in here, because they have to store a lot of starch, they will turn to another name that called grana chloroplast. That means no grana anymore, have only starch in them. And this grana chloroplast will be stay in the bundle sheet cell. I don't think I have a time to explain more about the structure of the leaf. So just go, it's your homework to study by yourself, okay? Look at the structure of the leaf. There are two kinds, C3 and C4 plant. Still remember that? C3 and C4. Familiar? Okay. Or if you are not familiar with it, just write down. Okay, C3 and C4. This one is a C4 plant. So after the epidermis, you will see the middle of the cell of the leaf. That's called mesophyll. The mesophyll have a lot of grana chloroplasts that used for photosynthesis. But the legs of the mesophyll, they have the bundle sheath cell that normally go around the xylem and phloem. Okay, and this bundle sheath cell will have a grana chloroplast that store starch for it. That happened only in the C4 plant. Okay, your homework. And here is the mitochondria that look like chloroplast, right? They have two layers, outer and inner membrane, but the inner membrane of the mitochondria will fold like this. So this fold we call cristae. This word, okay? The stick up of them we call cristae. Why it have to fold? Perfect. For what? Huh? For, yeah, to produce more energy. They have the ATP synthase. It's an enzyme inside the inner membrane here. So if they increase much more membrane, right? Membrane area, they will have more enzyme to embed it in. So they can produce more ATP. Good. And can you see both of the, oh, you cannot see in the chloroplast, yeah, but you can see in the mitochondria that they have ribosome and the circular DNA inside of them. But look at the chloroplast, they have two, ribosome and so-called circular DNA. They believe that both of them used to be bacteria before. And then they were eaten by the bigger bacteria. Yeah. And they just stay with the symbiosis together. So the chloroplast produce the food for the plant, right? And then mitochondria produce ATP for that cell. So they live together like that. And they can replicate themselves because they have own ribosome. They have own DNA. So they can replicate more according to the environment. Just like why the heart, the liver have a lot of mitochondria because it can reproduce. Yeah, many of amount of them in there. Another one is a vacuole. Vacuole, you know that it's used to storage, right? Used for storage. If they storage water, they will call contractile vacuole. Okay, just like in paramecium. Here is the water. When it's full of water, it will be like this, circle shape. And when it wants to move, so it will squeeze that contractile vacuole. If they squeeze the water, they can just move forward. Yeah, so that's how it works in the water. And if you have the protein inside, they will call food vacuole instead. So this vacuole can store many things. Can store the salt, can store pigment too. The pigment have to be dissolved or soluble in water because the vacuole is water, right? So the pigment that can dissolve in there have to be soluble in water, just like anthocyanin. Can chlorophyll dissolve in water? Just a bit. When you just boil the plant, boil the vegetable, it turns green, right? But it's not that green, dark green, because some of the chlorophyll get out in your water, in that curry. But how can you extract the chlorophyll? Normally we use the solvent that called petroleum ether. Have you ever heard? Yeah, petroleum ether, hexane, what else? Benzene, ethanol. In the market, we use ethanol to extract them. So if you buy the drink of the chlorophyll, you can smell a bit of alcohol inside because they extract with alcohol and then try to evaporate it out, but not all of them, and then mix with water later. Yeah, to save the cost. But alcohol is not the best solvent to extract them. Yeah, you have to use a non-polar compound. Have to use the non-polar solvent. Ethanol is very polar, right? Have the alcohol, the OH group, hydroxy group in there. That's why it can mix with water easily because it's a polar. And it can store the metabolic waste also. That's called calcium oxalate. Yeah. Calcium oxalate is in the plant that when you eat, it will irritate all of your mouth and skin. Yeah, you can destroy them. In Thailand, we eat a lot of this plant. Why, right? Why? Because it's fun to eat. Danger, but delicious. So you have to destroy them by the acid. Yeah, they in the frog medicine or in the frog way. So they use the lemon. Yeah, just put on them and then boil them. So you can destroy all of that calcium oxalate. Yeah, if you not burn your mouth. So if you heard about the news that people pick up the plant from the forest and eat them and everything is burned and go to hospital. Yeah, mostly that's from calcium oxalate. So don't eat things easily. Yeah, be careful. In Thailand, we eat a lot of dangerous things. You have to know the way of eating. Here is a plant. You can see that the vacuole is very huge, right? Because it storage many things in there: water, calcium oxalate, and chloride, sodium ion, anthocyanin also. What is the tonoplast again? Remember? What is the tonoplast? It has one layer of membrane. So you can say that it's a membrane of vacuole. Okay, vacuole has only one layer of membrane. Okay, another one that has one layer too. Here is a one layer of membrane organelle we call lysosome. Lysosome, lyso from lysis. Lysis means digest. So it will digest everything in the world. If you put the food in there, it will digest by the enzyme. So it will destroy all the food vacuole here. We call phagocytosis. If the cell just eat the food, right? Normally the food from outside, you have to bound with the membrane of the cell first, and then get inside as a food vacuole. We call food vacuole. And then the lysosome will come together and fuse with that food vacuole and digest by the enzyme. So you can see here is the hydrolytic enzyme. It is food particles already. How many enzymes in here? They said it's more than 100. More than 100 enzymes in one of the lysosome. And another thing we call autophagy. Autophagy means the breaking down of the damaged organelle. The lysosome will destroy the damage of the mitochondria. But can you see the mitochondria have to be fused with the membrane before? So it have to come in the vesicle form. And then they will fill with the rhizosome here and destroy them. Yes. Here is a real picture that the peroxisome fragment and the mitochondria fragment just filled with the lysosome. So they were destroyed together. Okay. Almost the last one. I know it's too much information. Yeah. Other of microbodies. These are the organelles that not many people mention about them, but you have to know them also, that we call peroxisome and glyoxysome. They both are digest the lipids, but it happens in only some of the cells. For the glyoxysome, it happens only in the plant. Yeah, in the germinating seed. The seed have a lot of starch in there and have a lot of lipid too. If it's monocot, just like the corn, right? They have a lot of starch. But if it's dicot, just like the peanut, they have a lot of lipid in there. So that's the difference between the monocot and the dicot, right? Storage starch or storage the lipid. Okay, so this germinating seed will have a lot of fatty acid inside. So the glyoxysome will break down all of those fatty acids. We break down all of those lipids into a fatty acid, sorry. So you can get energy from that. The plant can get energy from that breaking down process. And what about the peroxisome? The peroxisome happens in all eukaryotic cells. Okay, it used to destroy the lipid, but it's kind of long chain of lipid into a fatty acid. Yeah, and it's very useful to destroy the alcohol and harmful compounds also. So you can see a lot of peroxisome in the liver. Yeah. So the function of them are just like the RER, isn't it? So they work together between the RER and peroxisome. The last one. Last one. Yeah. Last one is about centriole. Centriole, you can see it only in animal cells. Centriole, it have the microtubule that connect together like this. But if we have two pieces that connect together all the time, yeah. Around them, we call centrosome. Okay, it's just like the area of the centriole. We call centrosome. But that centrosome contains a lot of compounds too. So what is the function of the centrosome? It's used for cell division, mainly for cell division. Okay, so all of these things you can read by yourself. It doesn't matter. Okay, it's a detail that I want you to understand more about this centriole, that it doesn't work easily, just like to produce the mitotic spindle and then pull the cell, just like that. It has the mother cell, daughter cell in there, and a lot of subunits in there that connect together. But you have to know that it's used for cell division. Yeah. And in other animals or other parasites, they have the cilia and fatula that contain microtubule too. I write down this. For eukaryote, centriole, we count a number as 9 plus 0 because here is around of the 9 groups of the microtubule. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9 is a 9 group of centriole. But the inside doesn't have any microtubule in there. That's why they call 9 plus 0. But for other animals, just like Flagellum, they have 9 plus 2 structure. 9 plus 2 means 9 group around them and outside, but other 2 inside. Okay. Here is the flagellum. If you cut the flagellum here, it will show 9 plus 0 plus 2. But at the basal body means where at the flagellum attached to the cell. This is like the base of them. So it will be 9 plus 0, 2. Just like the centriole. So everything that's inside the cell will be 9 plus 0. Everything that sticks outside the cell will be 9 plus 2. Okay. Like you need a stronger from the inner core, right? Because it's outside of your body and you have to move also. But you need a flexible. So you reduce the number of the microtubule out. It's like 2 of them attached together for one group. Not 3 of them anymore. Here is 3 of them attached together. Yeah, so this one is a stronger from inside, but still flexible. That's for movement. And here for the smaller of the microtubule that outside some of the organelles, or some of the organism. So then we have the cilia that's smaller, but a lot. Okay, and here is the summary of each organelle, that how many membranes that they have, what is the component of them, what is the function of them, that I group it for you. So you can read it by yourself if you forget. Okay, but I will upload the video and send a link to you again. Okay, do you have any question before we finish the class, student? The exam of this topic will be the function of each organelle, of course. Yeah. So you just match between the plant and organelles that exist in only plant or exist in only animal, and then the function of. So the number of the membrane doesn't matter, right? But if some of organelle have to use the inner side of membrane or the outside of membrane to have the function of them. So that's very important that you have to know. And the word, just like central dogma, what is central dogma? Yeah. So you have to know. So what is the endomembrane system? So you have to know, right? The specific word, right, that we have to know in the scientific term. Okay, see you next Monday with the cellular respiration. It will be harder than this. Good luck. Bye-bye.